All right, in this video, let's talk about the other three groups. In the last video, we talked about proteins. They're made of amino acids. Amino acids are the monomers, and polypeptides are, that's the umbrella term for the polymers, polypeptide. We also call them proteins and sometimes just peptides if they're short. Nucleic acids as well have monomers and polymers, and so do carbohydrates. And then we'll also talk about lipids, which is the only one of our four big categories of macromolecules that does not have a monomer or polymers. Okay, so nucleic acids are DNA and RNA. DNA is the genetic material. It's an acronym that stands for deoxyribonucleic acid, and RNA is also an acronym for ribonucleic acid, and that's why they are always capitalized. All three letters are always capitalized no matter where they are in a sentence because they are acronyms. DNA is the genetic material, which you probably already know that, and RNA is extremely important. RNA is how our genes get expressed. So when a gene is turned on, what is made first is RNA. And the monomers for nucleic acids are nucleotides. And nucleotides are a bit more complex in structure than amino acids are, or the monomers for carbohydrates, which are sugars. There's three parts to a nucleotide. One is a sugar, a 5-carbon sugar, either ribose or deoxyribose, which differ only in one oxygen. And it's shown as a ring here because sugars, which are a linear chain of carbons with a lot of oxygens on them, they tend to roll up into a ring. So glucose, fructose, and ribose tend to roll up into a ring structure. So that's why it's shown as a ring here. And the carbons are numbered, and that's what these little green numbers are. First, second, third, fourth, and fifth carbon. Ribose is a 5-carbon sugar. There's a base, and there are 4 different bases for DNA and RNA, and this is where we get the letters that you associate with DNA. The A, C, G, and T, which are the letters of a DNA sequence, which we could see in the title of that movie, Gattaca, which was using those letters. So those letters stand for the bases that are found on the 4 different kinds of nucleotides that make up DNA. And we're just going to use the letters. We're not going to worry about the names of them, but they are adenine, guanine, cytosine, and thymine. And then RNA has one different base. Instead of thymine, it has uracil. So for RNA, the 4 letters are A, G, C, and U, and there's no T in RNA. Why RNA has uracil and not thymine, we don't know. As you can see, thymine and uracil are almost identical. Just one methyl group differentiates them. And then the third part of a nucleotide is the phosphates, and this is an extremely important part of nucleotides. Nucleotides can have 1, 2, or 3 phosphates, even sometimes more than 3, but 1, 2, or 3 phosphates is the typical amount that we would see. If you take away all the phosphates, then we call it a nucleoside with an S, but we're not going to be dealing with that. But depending on what you're reading, you might see that word and think maybe it's a typo. So our monomers for DNA and RNA are nucleotides that have all 3 of these parts: the sugar, the base, and the phosphate. And it's the sugar that determines whether it's DNA or RNA. If that sugar is ribose, then it is RNA, and if it's missing one of the oxygens that's found in ribose on carbon number 2 here, then it is deoxyribose, and that makes that a DNA nucleotide. So we group these bases into groups based on whether they have a single ring or a double ring, and this is going to make a difference as to how they hydrogen bond together. As we'll see, a double ring base will hydrogen bond with a single ring base, and adenine can hydrogen bond with thymine. So in a DNA double helix, A and T will always be across from each other, and also C and G will always be across from each other, and that gives DNA a constant diameter. So the general term for DNA and RNA is polynucleotide because it's many nucleotides. Nucleotide is the monomer. Polynucleotides is the general term for the polymer, and the only polynucleotides that we find in living things are DNA and RNA. So here is our single polynucleotide chain, and the little purple shapes here, these are the sugars, and then the orange ones are the bases. So our backbone that's covalently bonded together goes sugar, phosphate, and you'll notice this P with a circle around it, that's a common chemical symbol for phosphate, which is a phosphorus with 3, 4, or 5 oxygens around it. So sugar, phosphate, covalently bonded backbone. The bases hang off of that. You remember these lines mean a covalent bond of shared electrons. And then these bases can hydrogen bond with another strand or other things. And these DNA and RNA are both extremely hydrophilic because phosphates are hydrophilic, the sugars are hydrophilic, and the bases are also hydrophilic. So the entire molecule is completely hydrophilic, which is a good thing to have as your basis for heredity in a living system. So DNA tends to occur as a double helix, which I'm sure you've heard that term before, DNA, the double helix, because there are 2 strands, both with their phosphate sugar backbone, that hydrogen bond together. So the 2 strands, they make a ladder-like structure that twists, and that's why it's a double helix. A helix, that means it twists. But if you straightened out the twists, it would look like a ladder with 2 backbones that are covalently bonded together, and the rungs of the ladder are just hydrogen bonded together. And remember, hydrogen bonds are a weaker bond. Hydrogen bonds can be easily separated with heat, which is what we do during a PCR reaction, is heat up the sample, and that separates those hydrogen bonds. You might have heard that the COVID test is a PCR test, and we'll learn more what PCR is later in the semester. RNA tends to exist as a single strand, although there are some exceptions. There are some viruses that have double-stranded RNA as their genetic material. But again, remember, we don't consider viruses to be a living thing. For living things, RNA tends to be a single strand, although it can fold up on itself. So these bases can hydrogen bond with other parts of their own strand in the same way. C can bind with G and A can bind with U, just like it could if it was a double strand. The RNA is much more flexible and tends to fold up in 3-dimensional shapes. So despite the fact that there's very little difference between the 2, just 1 oxygen different in the sugar and 1 different base, U instead of T, they have completely different functions in the cell. So here's our DNA double helix, and it has a constant diameter thanks to the fact that the 2 bases that bind together, one is always a double ring base and one is always a single ring base. And this is an example of the kind of structures that RNA folds up into. tRNAs are part of the gene expression system. This is part of how a gene for a particular protein gets turned into the actual protein. And transfer RNA is one of those. It's a single strand of RNA that folds up onto itself in this 3-dimensional shape that helps the right amino acid get added to the next amino acid in our growing protein chain. And we'll talk more about that later in the semester. So A and T hydrogen bond together, and G and C hydrogen bond together, and this is because of where those bases and charged hydrogens are. So in order to have a hydrogen bond, you have to have a positively charged hydrogen on one atom sticking to a negatively charged nitrogen on the other atom. So only between A and T do these line up correctly, and also between G and C. If the other bases are lined up with each other, it's a mismatch. They cannot hydrogen bond. And hydrogen bonding is extremely dependent on distance. If the nuclei of the atoms are just a little bit too far apart, then hydrogen bonding cannot occur. So here's another way that we represent DNA with a space-filling model. And you can see the nice twist there in the double helix. And again, this represents the covalently bonded backbone. So when we just heat it up, this backbone does not come apart because it's covalently bonded together, but heating it up does disrupt the hydrogen bonds. And you could also disrupt hydrogen bonds by changing the pH. That will also break them apart because hydrogen bonds are much weaker than the covalent bonds. The people who discovered the structure of DNA, once DNA was proven to be the genetic material around 1950, many groups of scientists tried to figure out the structure. And in England, we had Rosalind Franklin, using her X-ray crystallography, figured out that DNA was a double helix and that the phosphates and the sugars were on the outside and the bases were on the inside. And then Watson and Crick took that information and figured out the exact structure where every atom must be. And they got the Nobel Prize for their discovery. And Rosalind Franklin did not, because she had died of cancer by the time the Nobel Prize was awarded, and it used to be the case that if you died, you couldn't receive a Nobel Prize. However, that law, or that, not a law, that rule of the Nobel Committee was changed in 1972, and now Nobels can be awarded to people posthumously after they died. And so at any point since 1972, Rosalind Franklin could have been given the Nobel Prize for her work discovering the structure of DNA, and she has not. Despite an avid letter writing campaign by thousands of scientists, they have not corrected their mistake yet. So feel free to add to that writing campaign if you wish. Another function of nucleotides other than forming DNA or RNA is energy storage. Some RNA nucleotides are used for energy, and ATP is one that you may have heard of if you're interested in sports or nutrition. You might have heard of ATP. It is an RNA nucleotide. It's the A RNA nucleotide. It is the most commonly used source of short-term energy in our cells. It cannot be stored in large quantities. So we'll talk about carbohydrates and lipids, who also have an energy storage function. This is a little packet of energy that can be used by your muscles for muscle contraction, by enzymes to make reactions go more quickly or at all. And there's another nucleotide, the G nucleotide in RNA is also used for energy, GTP, although ATP is used for more things than GTP is. So that's another function of nucleotides, is short-term energy storage. Millions of these little packets of energy are used in your cell every second, all of your cells. So, and we call it ATP because there's 3 phosphates, adenosine triphosphate. And when that little packet of energy is removed, it's actually the breaking of a bond of one of those phosphates. So that little packet of energy can be removed and used for muscle contraction or cell movement or synthesis, and then it can be restored. We can use your mitochondria in your cells can take ADP and add a phosphate back onto it using the energy from the food you eat and create ATP again. But it is an RNA nucleotide. Okay, so carbohydrates. The name itself comes from the fact that carbohydrates have a basic molecular formula where they have, it's like you took the formula for water, H2O, and added a carbon to it, and that's the basic molecular formula for most carbohydrates. And here is an example: glucose, the most commonly used sugar in our bodies, C6H12O6. So if you divide all these by 6, you get CH2O. So that's why they're called carbohydrates, because that's the basic function. So in carbohydrates, usually every single carbon has an oxygen bound to it. The red here is oxygen and the dark gray is carbon. So every carbon has at least one bond to an oxygen, and that's typical for carbohydrates. And in fact, that's why carbohydrates are so hydrophilic and water soluble, because they have so many oxygens. And the bonds that store energy, everybody knows carbohydrates are for energy. You can use them, we can burn them for energy. The bond that stores the energy isn't the bond to the oxygen, however, it's all the other bonds. So the carbon to hydrogen bonds store a lot of energy, and the carbon to carbon bonds store energy. And those bonds are broken one by one, and that little packet of energy is harvested and used to make ATP. And that's how we harvest the energy from these. We're actually harvesting the energy from the electrons in that bond. So carbon to hydrogen bonds and carbon to carbon bonds, those electrons are at a high energy state, and that energy can be harvested and used to make ATP. Simple carbohydrates are sugars. You probably heard those terms as well. Simple carbohydrates, complex carbohydrates. So simple carbohydrates are sugars, and sugar, this is glucose, and you can see that there's a oxygen bound to every carbon, and there's one double bonded oxygen. Where this double bonded oxygen is determines which sugar it is. There are 3 6-carbon sugars that are really common. Ribose is a 5-carbon sugar. Glucose and fructose and galactose are 6-carbon sugars, and they only vary in the arrangement of the oxygens to the carbons. And sugars tend to roll up into a ring, so there's glucose rolled up into a ring. So glucose is by far the most common in our bodies. Every cell in your body can use glucose. The level of glucose in your blood is tightly regulated. If you don't eat for a long time, your liver makes more and dumps it out into the bloodstream. Fructose is much more rare. Fructose is made in tiny, tiny amounts as part of synthetic pathways. It's very hard for fructose to be metabolized. Most of the cells in your body, pretty much everywhere except your liver, can't really do anything with fructose. The fructose we eat has to be metabolized in the liver, and your liver mostly does one thing with it, and that is it turns it into fat. So that's why a very high fructose diet has been shown to be harmful, especially to the liver. Fructose, in fact, fructose metabolism has a lot in common with alcohol metabolism, and a lot of the side effects are the same. Fatty liver disease leading to cirrhosis of the liver, which is common in excess alcohol consumption, is also common with excess fructose consumption. And in fact, fatty liver disease, which is step 1 in cirrhosis of the liver, is really common right now in the United States, and it used to be only found in alcoholics, and now it's found even in children because of the absolute excess amount of fructose in our modern diet. And you might guess where fructose was first discovered just by the name. It has fruit in the name almost, and that's where fructose was first discovered. In fact, it's found in all plant sugars, which we'll talk more about the disaccharides like sucrose. Galactose, you might be able to tell from the name as well where it was first discovered. It's part of milk sugar. So lactose is milk sugar. It's actually 2 sugars stuck together. One of them is galactose. Galactose is extremely easy for our bodies to take care of as well. It's quickly turned into either glucose or stored as glycogen, and it's not a problem to consume large amounts of it as long as you can digest the lactose. So fructose, however, is in small amounts. Fructose is totally fine. It's the extremely high amounts of fructose that are very commonly consumed now, only in the last 30 years or so, that's the problem. So for sugars, we have a second type of group. So not just for the other groups, for proteins, we have monomers and polymers. For nucleic acids, we have monomers and polymers. For sugars, though, we also have a name for two sugars. So the Greek word for sugar is saccharin, or saccharide here. So mono means one, so a monosaccharide are all those single sugars that I just showed you. Fructose, glucose, and galactose are monosaccharides. So a polysaccharide would be many sugars stuck together. We also have a word for just two sugars stuck together, and these are still considered sugars, but they're so common that we have a special category for them. So disaccharides are sugars that have two simple sugars stuck together. And the most common one is sucrose. Sucrose is from plants, so pretty much any sugar from plants is sucrose. So whether it's beet sugar or sugar cane sugar or corn syrup, it's mostly sucrose. And sucrose is one glucose and one fructose. So when it's digested, and it is digested extremely quickly, digestion starts in your mouth when you eat sucrose. It's broken down into glucose and fructose, which, remember, have to be handled differently. So that means that sucrose is 50% fructose. So what's high fructose corn syrup? It just has sucrose with a little extra fructose. So high fructose corn syrup is usually 55% fructose instead of 50% fructose. So regular corn syrup or any plant sugar is mostly sucrose, which means it's about half fructose. So high fructose corn syrup is a little bit higher in fructose. Why would anybody use that? Why would food manufacturers use high fructose corn syrup? Because it's sweeter, so they can use less of it. And it's also cheap. So disaccharides are important in both animals and in plants because we say they're more stable. Not actually chemically more stable, but because cells that lack the right enzyme to break them into their two monosaccharides can't use them. So they're good for storage. So in plants, this is sugarcane here, and beets do the same thing. They're packed with sucrose, and the cells, the plant cells themselves that surround the storage regions, can't digest it because they don't have the right enzyme. So it's a good temporary storage. It keeps that glucose and fructose away from the metabolism and the metabolic activities of the surrounding plant cells. And the same goes for milk sugar. So lactose is a disaccharide made of one glucose and one galactose. And it's stored, the lactose is made by all mammals and is put into the milk. And because it's a disaccharide, that means that the mammary tissue that's storing the milk cannot use it for fuel. They cannot digest it. They don't have the right enzyme. And so it stays in the milk and is not absorbed by the surrounding cells. And then it goes to the baby mammal, whatever kind of mammal it is, whether it's you're talking about a human or a goat or whatever else. So, and the reason that lactose has become anything of concern is because most humans, by the time they're an adult, they've lost the ability to digest lactose. Their gut no longer makes the right enzyme, lactase, to break the bond between the glucose and the galactose. And if that happens, if undigested lactose makes it into your small intestine, completely undigested, and it does not get digested, it goes to the small intestine bacteria and then to your colon, where the bacteria just love lactose and they make lots of gas and absorb water and cause gas and diarrhea because of it. There are small populations that have acquired through natural selection the ability to digest lactose as adults. So if you have ancestors who are from Central Europe, who are from Sub-Saharan Africa, specifically the Maasai population, part of your ancestry, or from Mongolia. So there's 3 separate regions in the world where dairying cultures evolved. in the just recently, in the last 10,000 years. That's a blink of an eye in human evolution. So if your ancestors are from one of those three regions, you might be able to digest lactose as an adult. And sometimes, even if you can digest it as a young adult, many people, even who have ancestors from these dairying cultures, by the time they get to become elderly, they have lost the ability to digest lactose. So it's not just because you can digest lactose in your 20s doesn't mean you're going to be able to digest lactose in your 60s. So it's very interesting. The ability to digest lactose as an adult is a very interesting, recently evolved human trait. And it's a minority. Less than 50% of humans alive today can do it. Complex carbohydrates are long chains of sugars, and these are the polysaccharides. And that sugar is almost always glucose. So when you hear any kind of a complex carbohydrate, think glucose, because it's most likely 100% glucose. They have two main purposes. One is structural, and the other is for energy storage. So if it's a structural carbohydrate, like in a cell wall, that means it cannot be used by the organism that made it for energy. So we'll talk about some examples of that. And we'll talk about energy storage carbohydrates as well. So for plants, they store, when they're not storing sugar temporarily as sucrose, they can store it longer term as starch. And there are many different kinds of starch. Here are just two of them, amylose and amylopectin. There's many, many different kinds of starch. What a starch is, though, is a polysaccharide of 100% glucose. Some of them we can digest really easily, like amylose. We digest extremely easily. Amylose is the major starch that's found in potatoes. But there are starches that are found in beans, for example, that are still 100% glucose, but the way that they're linked together, we don't have the right enzyme to break them apart. But you have bacteria in your gut that do. So those starches make it to your large intestine completely undigested, and the bacteria break them down and make lots of gas in the process. So starch is just glucose, and this is why if you're a diabetic or you have a family member who is, you probably know starchy foods like bread and rice and potatoes really raise your blood glucose because that's what they're made out of. They're 100% glucose, and they digest very quickly. In our bodies, we also store glucose in a large polysaccharide, but in animals, it's the glucose molecules are put together differently, and it's glycogen. And of course, we can digest glycogen very easily if we consume it. It's found in small amounts in certain foods, like if you eat beef liver or if you consume meat of any sort. There's small amounts of glycogen in there, and we can digest that very, very easily because, of course, our bodies make glycogen. And most of your glycogen is in your muscles and in your liver. The liver is in charge of regulating blood glucose along with your pancreas, and if your blood glucose gets low, the liver breaks down glycogen and dumps glucose into your bloodstream. So that's how, if you're, prepping for surgery and you don't eat for a whole day, and then you don't eat on the day of the surgery, but your blood glucose is going to remain stable because your liver is breaking down liver glycogen and dumping it into your bloodstream. And when that runs out, your liver also has the ability to make glucose, which is called gluconeogenesis. And it's able to maintain it can make glucose from fat if you completely run out of your glycogen stores. Glycogen is stored in your muscles for immediate use, and this is what runs out when people say they hit the wall when they're running a marathon, and around mile 20, most people hit the wall, and that means that all your glycogen stores are now gone, and you're running completely on your fat stores, which are slower to turn into glucose. It's a much longer, slower process to turn fat into glucose than it is to turn glycogen into glucose. So when your glycogen is all gone, you become very fatigued, and that's the hitting the wall phenomena. So structural carbohydrates. So this means that for the organism that's making it, it is not available for energy. It's not an energy storage. It's a structure, and it might be eaten by something else that can use it for energy, but that organism itself can't use it for energy. So plants have a structural carbohydrate called cellulose, and this is what plants make their cell walls out of. We call it fiber because we can't digest it. So if you eat cellulose, and if you eat any plant, you're eating cellulose. There's some cellulose in all plants, and it passes through our system without being digested. We lack the correct enzymes to break down cellulose. The only organisms that make the right enzyme to break it down are certain bacteria and fungi can break it down. And that's why animals that can break down cellulose in their gut do so thanks to symbiotic bacteria that live in your gut. We don't have the right kind of gut to host these particular bacteria, so we don't get any of the glucose in cellulose. So cellulose, again, is made of 100% glucose, but that energy is unavailable to us because we don't have the right kind of gut. But a cow does. So many animals, cows, goats, sheep, have fermenting guts. Most primates have, other primates other than us, we're primates. Most of the other primates, like even chimpanzees and gorillas, have some gut fermentation. And so what that means is cows have a very advanced kind of gut fermentation. So they have bacteria in their gut that break down all the cellulose, release all the little glucose molecules, and then the bacteria use that glucose to grow. And then that moves down their gut, and the cows are really eating the bacteria. So all those bacteria that they feed with all the plants that they eat, those bacteria turn that glucose into protein and fat, and that's what the cows eat. So we don't have that ability. We cannot digest cellulose. It passes through our guts undigested. There is one group of animals that have a structural carbohydrate, and those are the arthropods. So here's a little shrimp here, a mantis shrimp. So the arthropods are everything that has a hard exoskeleton and jointed legs. So that includes all the insects, bees, wasps, ants. Those are all insects. Crustaceans would be things like shrimp and lobster. Those are all arthropods. Crabs are arthropods. So all that hard shell on a crab or a shrimp or a lobster or a spider is made out of carbohydrate. It's made out of glucose, and it's a structural carbohydrate called chitin. And interestingly, fungi also use chitin in their cell walls. And here's one of the links between animals and fungi. If you remember way back from the first lecture, the closest kingdom to the animal kingdom in evolutionary terms is the fungal kingdom, and here's one of those links. Chitin is made by some animals, and it's also made by fungi. So, and again, we cannot digest chitin. Some animals can. Animals that eat a lot of crustaceans or insects can digest chitin. Sometimes we don't, though. If you eat shrimp shells or I don't know why you'd eat a lobster shell, but I know sometimes people eat shrimp shells if they're deep fried. It won't hurt you, but you also won't get any calories from it because those little glucose molecules stay together as they pass through our digestive system, and we don't have any gut bacteria that can break them down either. Chitin has been used for many things. It's extremely strong, and it's been used as a dissolving surgical thread. You might know that if they do internal stitches on you during surgery, they don't want to have to open you up again to take the stitches out, so they use various things to make stitches that will slowly break down in your body, and chitin is one of the things that they make them out of. They used to make them exclusively out of animal intestines, which was called cat gut, even though it wasn't really from cats. But now they mostly replace it with synthetic things, and even synthetic chitin is used, because even though we can't digest it, we don't have the right enzymes to break it down. If it's in the form of stitches inside your body will very slowly break it down over weeks. Lipids, our last group. So lipids are unlike the other three groups because there's no monomers and polymers. So for proteins, we had amino acid is the monomer. Polypeptide is the umbrella term for all the polymers. For carbohydrates, we have sugars, and monosaccharides would be the scientific term for sugars. And then we have polysaccharides, or the complex carbohydrates. Monomers and polymers. And then nucleic acids, we had nucleotides, and then we have DNA and RNA, or polynucleotides. So lipids, we don't have that monomer and polymer relationship. In fact, lipids are kind of a catch-all category. So because our cells are a watery environment, inside the cells is a watery environment, outside the cells is a watery environment for all living cells. Therefore, almost everything is hydrophilic. That's an organic molecule. Proteins, carbohydrates, nucleotides, they're all hydrophilic. The things that are not hydrophilic, because there's fewer of them, we lump them all together into one category that we call lipids. And lipids, so we're going to talk about the three major categories of lipids. This would be fats or triglycerides, which you probably heard of triglycerides in terms of that's one of the blood tests that they do on people, is what level of triglycerides do you have floating around in your blood. The sterols, which are based on the cholesterol molecule. Cholesterol is an important molecule that's in membranes, and also we make a lot of hormones from it. We'll talk about those, and also phospholipids, which is what membranes are made out of. Every membrane from bacteria, archaea, and all eukaryotes, the membranes are made out of phospholipids, so they're very important. So triglycerides, which are also just called fats. So lipids and fats are not synonyms. Lipid includes more different kinds of molecules. Lipids just means everything that's hydrophobic that we find in cells. But fat refers to something very specific. A fat is a triglyceride. So we have fatty acids, and you've probably heard that term before, fatty acids. There are dozens of different fatty acids that you find in animal bodies. And fatty acids are long hydrocarbon chains, so you see nothing here that's going to build up a charge, nothing here that's electronegative. We just have carbons and hydrogens. So fatty acids are a long chain of just carbons and hydrogens with one carboxylic acid group on the end. These are hydrophobic. This one little carboxylic acid group on the end is not enough to make it hydrophilic. So that's a fatty acid. Fatty acids are inconvenient for energy storage, though. They are not as stable as they could be because of this carboxylic acid at the end. So our bodies and plants do the same thing. All animals do this. We link them together to make a molecule that has 3 fatty acids. And the thing that links them together is this weird little sugar. So this is a 3-carbon sugar. How can we tell that it's a sugar? Because every carbon has an oxygen, and there's no other elements. It's just carbon, hydrogen, oxygen. This is a sugar. So it's a 3-carbon sugar that's called glycerol. And glycerol is a really important molecule. It's a really important metabolic intermediate. It's important in photosynthesis and in cellular respiration. So anyway, this little 3-carbon sugar gets 3 fatty acids linked to it, and there's our triglyceride. And that's because the glyceride, that word comes from the glycerol here. So it's 3 fatty acids linked to a glycerol. And they don't have to be all the same. There could be 3 different fatty acids, or there could be 2 of one fatty acid and a third different fatty acid. And the fatty acids are different because of how long they are. So there are 8, 10, 12, 18, 20, 22 carbon long fatty acids. It's always an even number of carbons. And they also vary in whether any of these carbons have double bonds. So those are the 2 ways that fatty acids vary. But they never have any other elements in here. There's never nitrogen or sulfur or anything. So fat is triglyceride, and it doesn't matter if it's a liquid or a solid at room temperature, it's still a triglyceride. So whether you're talking about butter, lard, suet, olive oil, corn oil, they're all triglycerides. It's the difference in whether it's a solid or a liquid at room temperature is which fatty acids are present. And if the fatty acids have a lot of double bonds, that pushes those fatty acids further apart and makes it more liquidy. So if you have something that's solid at room temperature like butter, that tells you that there's less unsaturated fats and more saturated fats. Now, nothing, unless it's made in a factory, is pure saturated or unsaturated. It's always a mixture. So butter has saturated fats, monounsaturated fats, and polyunsaturated fats. It just has more saturated fats, so it's solid at room temperature. So what does that saturated mean? That has to do with these double bonds. So any double bond, even if there's just one double bond, that is considered an unsaturated fatty acid. So, and that term comes from chemistry, that if it has a double bond, we can force more hydrogens in there. So it's not saturated with hydrogen. So that's why it's called unsaturated. A saturated fat has no double bonds, and that means it's saturated with hydrogen. We can't force any more hydrogens in there because there aren't any double bonds. So saturated means no double bonds. Unsaturated means there's at least one double bond. So monounsaturated, mono means one. So a monounsaturated fat has one double bond. A polyunsaturated fat means there's at least two double bonds in the fatty acid. So, and the structure is different. So for a natural unsaturated fat, like olive oil, oleic acid is a monounsaturated fat, meaning it has one double bond. It's found in olive oil. It's also found in animal fat, like beef fat. You find oleic acid in there as well. And butter has oleic acid in it. It only has one double bond, and the structure of a double bond in a natural fat is going to be what we call a cis double bond. Now, we use these terms cis and trans in biology and in chemistry, and have for decades. In chemistry and in biology, cis means close together, and trans means away from or further apart. So in a cis double bond, the two next carbons are pointing in the same direction. So we have our double bond, and the two next carbons are pointing in the same direction. That's a cis double bond, and that's what most natural unsaturated fats have, and that's what makes them liquidy. So if your oil has a lot of oleic acid, monounsaturated fat like olive oil, at room temperature it's going to be a liquid. But if you put it in the fridge, it should turn into a solid. And this is a good way to test your olive oil. If you want to know if your olive oil is mostly real olive oil, because there's a real problem in the olive oil industry right now with people adulterating olive oil with soybean oil and corn oil and adding a little green dye to it and selling it as pure olive oil. If you put your olive oil in the refrigerator and it stays a liquid, that means it's mostly soybean oil or corn oil or some other cheap vegetable oil, because real olive oil, when you put it in the refrigerator, it becomes a solid in the refrigerator, but it's liquid at room temperature. So do that test with your olive oil at home. When I've done this at home so far, I have found that all the California olive oils turn into a solid in the fridge, but I have found some imported olive oils that stay a liquid when I put them in the fridge, which means that they are not pure olive oil. Another type of fat that we hear about is trans fats, and we've been hearing a lot lately about trans fats are bad and don't eat trans fats, and they've actually been, food companies have been mandated to reduce the amount of trans fat. So what does that mean? So to make a trans fat, you need a factory. Polyunsaturated fats, so these are fats that have lots of double bonds, at least two in each fatty acid. You can artificially saturate some of those double bonds by heating them up and putting them under pressure. And this will blow some of the double bonds. And what you end up with are these trans fatty acids. So here's a fatty acid that has a double bond, but instead of, so here's our double bond, instead of having both of the next carbons pointing in the same direction, now they're pointing in opposite directions. And so this fat will behave more like a saturated fat. And why would you want that? Why would companies want to use these hydrogenated oils? It's because they behave more like saturated fats for cooking purposes. You might know that saturated fats, if you're going to fry foods, like if you're going to make homemade French fries or something, saturated fats like lard and beef tallow work better. That saturated fats have superior qualities for making fried chicken or other fried foods at home. And the same is true for industrially produced things that are fried, like crackers and things. And also they have a really long shelf life. These hydrogenated trans fats last much longer on the shelf. Saturated fats tend to break down quickly. Bacteria like to eat them. They become rancid. So if you're going to make cookies for the grocery store and you use real butter that has a lot of saturated fat, they're not going to have a very long shelf life, maybe a week, and they'll start to taste rancid. But if you use hydrogenated cottonseed oil, it will have a much longer shelf life, maybe as long as a year if you use that. And you will get the same kind of crispy properties that you would get with a saturated fat. So that's why they use them. And why are they bad? Trans fat consumption has been linked with heart disease, strokes, and other things. And it's now looking, though, like it's not just the trans fats, it's the polyunsaturated fats in general. That diets high in polyunsaturated fats, whether that's soybean oil or canola oil or any kind of vegetable oil, that it doesn't matter if it's hydrogenated into a trans fat or if it's in its natural polyunsaturated condition, that is itself associated with poor outcomes. And so organizations have been very quietly lifting their advice about avoiding saturated fats, because it turns out that saturated fats aren't the devils that we were told that they were. We also hear about omega-3 versus omega-6 fats, and this relates, they have to have a double bond to be an omega-3 or an omega-6 fat, because that tells us where the double bond is. It's just a chemical naming system. So in a fatty acid, the first carbon after the carboxylic acid is the alpha carbon. The last carbon in the chain is the omega carbon. You can see the little tiny Greek, lowercase Greek omega there. So if it's an omega-3 fatty acid, that tells us that the first double bond is 3 carbons in from the omega. So this last one here, linoleic acid, this is an omega-3, or linolenic acid rather, is an omega-3 fat because the first double bond is on the 3rd carbon in. So that's what that means. And again, just like with hydrogenated oils, the trans fats, omega-3 fats are correlated with good outcomes. Omega-6 fats are correlated with poorer outcomes. And this is why people are taking fish oil and eating salmon, and that is to get more omega-3. And there's actually quite a bit of evidence that there might be something to that in intervention studies where they give people more omega-3 fats. That they actually do better. So omega-3 fats are important. So anything that's hydrophobic is a lipid, and there are some weird things that then are considered lipids, like chlorophyll is actually in the lipid category because it has this long fatty tail that helps it stick in the membrane. Plants make a lot of weird lipids, they have their own steroids as well that we don't use, that are found in their membranes. But even things like rubber and natural latex is a plant product. It's also a lipid. And then we'll talk about phospholipids, which are what membranes are made out of. So steroid hormones are extremely important. Plants have their own steroid hormone as well. They're all made from cholesterol. Cholesterol is extremely important. You can make pretty much every cell in your body can make cholesterol. They make it if you need it. Cholesterol is very important to the function of your brain, to the function of your nerves, and also all of your steroid hormones depend on those cells' ability to make cholesterol to produce estrogen, testosterone, and progesterone, which are the sex steroid hormones. Men and women have all three, estrogen, progesterone, and testosterone, just in differing amounts, and they do different things. Corticosteroids are sometimes called the stress hormones. Cortisol and corticosterone, these are made in the adrenal cortex, which is above the kidney. Also extremely important, there are diseases associated with not having enough of these. If you have too much stress hormones, of course, that's not good either. But again, these are both made from cholesterol in the cells that produce these hormones. Cell membranes are made out of phospholipid, and the secret to success for cells is the membranes. Membranes are an effective barrier against the watery outside world because they provide a fatty barrier that hydrophilic things cannot cross. So every membrane has these phospholipids. And if we look here, a phospholipid is almost a triglyceride. We have our 3-carbon glycerol here, 2 fatty acids, and the fatty acids can vary enormously depending on what kind of an organism it is. And then that 3rd glycerol, instead of having another fatty acid, it has a phosphate, which is why they're called phospholipids. Phosphate is very hydrophilic, and then it has another molecule which varies again depending on the species, and usually has nitrogen in it, in this case choline. This is a common one found in human membranes. So we have half of the molecule that is hydrophilic and wants to stick in the watery environment inside or outside the cell. And then we have these hydrophobic tails. And remember that in water, things that are hydrophobic want to glob together. And that's how membranes stick together. Your cell membranes are not attached to each other. The molecules that make up the membrane, all these phospholipids, are not attached to each other by covalent bonds. They're just sticking together by hydrophobic interactions, one of the weakest interactions that we know of, but that's enough to maintain cell membrane integrity. So the little white things here are supposed to be phospholipids, and so you can see it's a double layer, and so every membrane is like this. In bacteria, all the way to every animal and plants, all the cell membranes are made this way. They're a double layer of phospholipids with that hydrophilic phosphate and nitrogen containing polar head facing out and another one facing in. So we have this long fatty region here that nothing hydrophilic can get across. Anything with a charge, ions, sodium ions, hydrogen ions, water itself cannot get across this fatty layer. And these little yellow things here, that's cholesterol. So cholesterol is a really important component of our membranes. It helps stabilize the membranes, it helps makes them flexible. Plants have their own sterols, not cholesterol. You probably know plants don't have any cholesterol in them. Any plant foods don't. But they have their own sterols as well. We just don't absorb them. They stay in your gut, hopefully, because they're not very healthy for us if they manage to get into your body. And then you see there's proteins stuck in the membrane here. Some of them are channels, like this one is going to allow, perhaps this is an aquaporin channel. But that's our effective barrier for everything in the cell, are the phospholipid bilayer. So for each of these, we'll practice in a discussion categorizing the monomers and polymers. So for proteins, the monomer is amino acids, and the 3 names we use for the polymers are proteins, peptides, and polypeptides, with polypeptide being the umbrella term. For nucleic acids, we have nucleotides as the monomer, and DNA and RNA are the polymers. For sugars, we have sugars are the monomers, and then we also had another category, disaccharides, for two sugars stuck together, and then polysaccharides for starches, cellulose, chitin, glycogen, all those large things that are made out of glucose. Lipids is a catch-all category, and we don't have monomers and polymers for lipids. We talked about fatty acids and triglycerides, phospholipids, which make up membranes, and things based on the cholesterol molecule, including cholesterol itself being part of membranes and all of the steroid hormones that are made from cholesterol. So that's it for the biologically important molecules. There are, again, other molecules that don't fit into one of these four categories that are important. Many of them are vitamins because they're important components of our cells, but they're in small amounts and they don't neatly fit into one of these categories. So there are other things, but these are the important ones. Okay. So that's the end of biochemistry.